What I really didn't want to do was solve the wrong problem, because that, it'd be so much worse. Working on the right problem and failing, you're still maybe one step away from succeeding. But if you're working on the wrong problem, it's a lot harder to navigate to making an impact.
This is the Nucleate Podcast, the voice of the next generation of biotech leaders.
Welcome to the Nucleate Podcast. My name is Sam Kessel, and I'll be your host. On this episode, I have a conversation with Eric Kelsic, the CEO and co-founder of Dyno Therapeutics. Prior to founding Dyno, Eric led a team to develop the technology underlying Dyno's artificial intelligence-powered Capsid engineering platform in George Church's lab at the Wyss Institute at Harvard Medical School. There he measured the first comprehensive fitness landscape of the adeno-associated virus, capsid protein, and co-discovered the AAV map gene. He earned a PhD in systems biology from Harvard University and a bachelor's in physics from Caltech. Under Eric's leadership, Dyno has raised over $100 million in venture financing, including a 2021 Series A led by A16Z, and has signed partnerships with world-leading gene therapy companies, including Roche, Sarepta, and Astellas. Dyno was named X-Economy's 2020 Startup of the Year, and Eric was recognized as one of the Endpoint's 20 under 40 next-gen biotech leaders in 2021. And on this episode, we cover broad topics across how AAV gene therapy works and how Dyno's platform has evolved to use machine learning and artificial intelligence to develop the next generation of capsids to ideally make novel therapeutics that reduce the cost for patients and make a transformative impact on disease. We hope you enjoy the episode. Thank you so much for joining us on the Nucleate Podcast today, Eric.
Thanks, Sam. Really happy to be here.
So let's start with your background. You have a unique context in biotech with your undergraduate studies in physics, as well as studying systems biology and data science. Can you tell us a little bit about how this lens has impacted your approach to biological problems at Dyno?
So someone once gave me some good advice on how to choose your career path, and that really stuck with me. And it's that you want to find a place where you can uniquely have a really exceptional impact. So you mentioned my background in physics as an undergrad, PhD in systems biology. There's like an intersection between those two, which provided me focus, and looking for the ways in which those unique skills could enable me to do something that only I could do. That's kind of how I thought about my next steps after my PhD. And then the other side of it is, what of those things am I really motivated by? So that further narrows it down. And then just kind of openly exploring around different ideas there. That's what ultimately led me to the problems that we're working on that became Dyno. Solving delivery, working at the cutting edge of different technologies, but all that comes from the background of the technology. But with that really specific focus on technology is going to help patients that are going to really transform lives. So it's finding that intersection that really helps me, and then keeping an open mind to explore around what problems would qualify for those characteristics.
Do you notice that, with your physics background or the systems biology approach, that you fundamentally assess problems differently than your colleagues who are, say, pure bio majors? Is there a different, first principles type of problem solving that you use in biology that's different because of your background?
Yes and no. I mean, everyone's going to start with what's familiar to them, and certainly I've worked on some problems that way, but on the other hand, that's not an excuse. So for example, coming from physics into biology, actually many great biologists have come from the physics background. So looking at the history, there's just so many great examples of impact. But one thing I would see pretty frequently when I first got into the systems biology field would be others who had come from other fields like physics, and they would say, oh, I'm just a physicist. So, so what do I know? I'm not a real biologist. And that always kind of bothered me because, it's an excuse, and it's kind of like holding you back if you let that limit you. So early on in my PhD, I really wanted to just dive in, not a physicist anymore. I'm a biologist. I've got this background that's going to enable me to find problems that maybe others wouldn't, or solve problems that others wouldn't, but I have to also round out my skill set so that I'm not missing some basic fact that's gonna lead me to waste time. So doing the things so that I no longer felt like I needed to disclaim that I came from that background, that was really where I spent a lot of time and focus in the early parts of my PhD. And then maybe midway through, I had done that, and I had done kind of um, more basic biology to really dive in. And then I started to ask, well, I could keep doing that, but I'm not going to necessarily contribute something that no one else would. So how can I use that background that I have in a more impactful way? Like, for example, physics with mathematical kind of reasoning, that was there, but I had also done a lot of programming. So it was really the kind of programming aspect that first let me realize, oh, I could approach this really simple experiment, but in a different way that uses programming. And then I could, design the experiment much differently, or design it in a way that I could analyze that experiment much differently than if I didn't have those skills. And that's something that I can do that I don't see others doing. So kind of following that thread and then seeing where it leads you can then reveal opportunities that others haven't seen before. And now you're the first, and now you've got a chance to, do something exciting.
Yeah, and I think a lot of innovation happens at the intersection of multiple fields, and that you can, to your point, still understand the key fundamentals of biology, but also speak a whole fundamentally separate language of physics, mathematical reasoning, and computer programming to apply a different lens to those problems, which I think is really fascinating. And obviously, we'll talk more about how you do that at Dyno. So you alluded to your PhD that you did at Harvard. You ultimately did a postdoc with the legendary George Church. So curious what lessons you learned about working with George Church, about his approach, his lab, and how you developed as an entrepreneur and scientist working with him.
Well, I learned so much from George and from his lab, and maybe that's another learning is finding ways to put yourself in the right environment where you can learn, even if you don't know exactly where you're gonna apply yourself, but well, how would I get to that point? That's what I was thinking when I asked George if I could join his lab, because I knew I wanted to go into industry. I knew I wanted to take the research I had done and apply it to something that was going to be impactful for human health. And I felt like George had created this environment that was had all the right components that would enable me to help George in the lab, but also learn what I needed to find the right problem and then go help a company. So what I learned from George is, he really thinks a lot about the people, and he's a great person to catalyze this community, to bring all the right people together, to give them the space to think openly, to push them at times to think bigger, or just to go a few steps further, and then opportunities arise from that. So that's a lot of things have to go right for that to happen, but George has really found a way to make that happen. And so when I joined the lab, there wasn't a specific thing I wanted to work on, but it was because of these things, there's probably something interesting here, good for the lab, good for the work that we're doing, trying to create technologies that are going to help people. And George was very open to that, even without knowing exactly where it would lead. And I think that doing that in all sorts of different ways, that's how you create this critical mass of ideas and opportunity and motivated people. And then, just starting with things that are not great ideas and turning them into great ideas with, pushing in different directions. I think giving an idea time to incubate, maybe. George is very good at that. He would never say, no, don't do that, right? Because every good idea maybe started as a bad idea, and then with a twist, it became a good idea. So the bad ideas, you can't reject them right away. And George is very good at not rejecting any idea, especially when it comes to technology, but more asking, well, what if, what would need to be true for this to be possible? Or that might be hard to do, but how can we get one step closer, and then think about that problem? And so there's just a certain set of questions that George is good at asking that start the conversation. And I think that's one reason why the lab has been so successful across so many different areas. It's a big lab, but they work on a lot of different things, and they often are. George is very good at being the first to do something. And then a lot of other people can build on that, but the first step is often, unblocking and catalyzing for all that future innovation.
It's really interesting that you mentioned that he didn't really say no to anything. It was more, how can we understand this idea better? Can we think about this idea from a different angle? Having that open-minded inquiry and not shutting down things. I think that sounds unique in a way, to have that open exploratory approach and not shooting people down. I think is great from, certainly from a scientific perspective, but also from the entrepreneurial perspective as well, is to not immediately shoot things down, be open-minded, and see where ideas may lead you.
Yeah, and I think George and I shared the value that I mentioned before around, is there something I can help with here? So I think that's how George thinks about it. Is there something a lab could do uniquely? Is there something that he could do? In some ways, the funnier the idea, maybe the more potential there is to contribute, given those, all the experience that George has or that the lab has. So he's really good at kind of finding the right people who want to work on something different or who want to try to do something big. And maybe they will be successful, maybe not, but they're more likely to be successful in the lab. I think that's a good way to think about it, and really a good way to think about any organization that wants to be innovative. Like how do you assess that, knowing that innovation is it's an unknown, so it's hard to know what's possible until you try. So there's a certain set of values to working together and an openness to making new connections, sharing information, collaborating. And George is also very good at that, connecting the right people. And then, these teams self-assemble and then they go off and focus for a year, for several years, and come up with something really amazing.
That's great. And that's a good segue to discussing, how you eventually started Dyno. Can you speak to how that all started? If there's any inspiration or learnings from your experience with George Church and how that manifested in how you started Dyno?
It's an iteration, so I can kind of talk about the successive steps of how that came about. For my PhD, one part of it, I had worked on high throughput experiments, mutating genes in E. coli, reading out the results with high throughput sequencing, actually in collaboration with a postdoc in George's lab. And from that, generated some of the first completely comprehensive fitness landscapes. And I could see patterns in the data. I spent a long time analyzing this data at the protein level, at the RNA level, to try to understand those patterns. But it was very clear to me that this data is very rich. And if there's patterns there, then you wouldn't need all of the data to be able to predict the rest. And if you could predict, then maybe you could engineer. So while my research as a PhD was more basic, the applications for protein engineering were apparent. And I wanted to find some way to explore that in a context that was going to be beneficial for human health. So that was my thought about eventually going into industry, working in a startup. And these technologies are going to be meaningful for that. So I joined George's lab thinking I would look for a problem that I could solve with that measurement of technology. George's lab also pioneered the DNA synthesis side. So it's not just reading DNA, but also writing DNA in high throughput so that you could test those predictions. So I wanted to learn that set of skills because that was going to be key to making use of the information. And then experience from my PhD of analyzing this big data set, the way it worked was it took about 3-4 months to develop the measurement technology, do a pilot, and then that was working. And so then in 1 week, I could generate huge amounts of data. So one experiment took a week to generate. It was something like 60, 70,000 mutants. And then I spent 2 years analyzing the data set to find out what happened. So in addition to reading the DNA, in addition to writing it to design new experiments, I needed some way to know how to design the experiment, and it needed to be shorter than 2 years. So this was right around the time that deep learning was coming into the mainstream. There were a lot of results from image analysis, speech analysis, showing that you could automatically learn representations. And so if that was true, then why shouldn't it also be true for protein sequences? That was my observation. And it became also much easier than, this is 2015, to do deep learning because there were frameworks like TensorFlow and cloud computing were making it easy for you to get these things up and running. So my thought was the kind of 3 technical pillars were going to be DNA sequencing to measure the results of experiments, machine learning to analyze them, and then DNA synthesis to test those predictions and ultimately to iterate through this approach. And I was looking for a problem where that would be relevant. And so the first few months of joining the lab were going around talking to different people. And then also I asked George to connect me with some lab alumni who are working in industry in different contexts. I talked to a person who worked at a genome editing company and, trying to understand what problems they saw. I saw a lab meeting presentation about delivering CRISPR editors using AAV in mice. And that was maybe the first time I saw the delivery problem, because it was a fluorescent reporter and a histology slide with one cell that was edited, which had changed color, and all around it were unedited cells. So it was very clear that in order for genome editing to be successful in vivo, that needed to be improved. And so that was the problem. And then I went to talk to folks in industry and asked, do you have this problem? And they all did. So this in vivo editing or even in vivo delivery became aware of that as a problem. And it seemed like everyone who was doing that was using the best available technology, which was AAV capsids, this protein shell that comes from natural viruses, but the natural viruses are not, the capsids are not good enough for a therapeutic application. So now I've got this protein engineering technology, this potential target, and I wanted to explore, is this the right application? So the companies that I were talking to, I would go talk to the AAV expert and ask them, and they would say, yeah, we need better AAV. That's for sure. And so what I ended up doing maybe 4 months in, 6 months in, was I took a course at the Harvard Business School, audited it, and worked with a team there, and kind of did it in reverse of how it's typically done. Typically, this course is the teams are paired with the technology from the tech transfer office, meaning like technology has already been developed, maybe already had applications for patents filed, and universities thinking about commercialization, looking for ways to commercialize that technology. The technology is already done. So for me, instead, I was pitching my idea for a project, which could become a technology. And the question is it worth it to do? So that meant really like building out the case for it, talking to a lot more different people in industry, including investors who had started companies, and asking what would it mean if we had solved this problem? And that really solidified my confidence that this was a problem worth solving. People have been working on it at that time almost 2 decades, and with not a lot of progress. So, actually one thing I learned from my PhD was when there's a hard problem that people haven't had made progress on, that's a good thing, because if you're developing a new approach or a new technology, you want to find something that people couldn't solve through another standard approach. So it was good that people agreed it was important, but had been trying to solve it and hadn't yet succeeded. And then I started thinking about it. Now that I know what I could do, I could go join one of these companies and help them with that. But since all the companies have the same problem, and since one AAV capsid could be used to deliver many different payloads, so in principle, one capsid could help many companies, maybe there's a different opportunity here. Maybe there's one company that could solve this problem and help all these companies, the whole industry. So my ultimate plan when I wanted to go into industry was join a startup, learn how they work, but I had consulted for some startups as a graduate student. So the reason why I liked that was I really liked the team aspect of it, like working together to solve a whole problem. And I thought that after I had that experience working at a company, someday I would have an idea and start a company of my own. And now this seemed like a really great idea that I happened to be like recognizing very early. So I thought maybe there won't be another chance like this. Maybe this is the idea, and changed my plan. Decided instead of going to join a company to do this, like I think I know how to do it. Looked around in the lab. Actually, we have the resources to test this. So let me at least take the first technical steps and understand whether this could work or not, and then decide if it's to pursue it as a spin-out company. So that changed my plan from what originally was a 6 to 9 month postdoc to what finally became almost 4 years of a postdoc, getting the technology working and then bringing it back to the same companies or to investors and asking, what do you think? So maybe like 6 months of evolution, realizing this was an opportunity, and then deciding the next step is to technically de-risk it, realizing that Georgia's lab was actually a great place to do that. Not only was I interested in startups, but many other people in the lab were working on ideas that could become startups. Several of those folks were further along, so I could learn from them. So it ended up being a really great place to learn, and then sticking with that, solving the technical problems, and then finally feeling the urgency to get out and build a team and to go faster.
Well, it's really interesting that you initially started with an insight from data, right? That you were finding these, potentially engineerable and predictable engineering approaches early on in your PhD, and you were really looking for how do I approach the right problem with this novel method. So I think that that's an interesting origin story, in that you did a lot of really validation of the problem, and you explored it with a bunch of different, sounds like, strategics, investors, etc., to validate, yeah, this is a big hairy problem. This is something that hasn't been solved for 20 years, and that this insight that you have on protein engineering can be applied to solve AAV gene delivery, and that this is something not only that is meaningful to solve a challenging problem, but it's a problem that matters to a lot of patients, right? To the point that you said from all along, that the focus from the beginning for you is, it's great to have a new technology, it's great to have data and insights, but is this solving a problem that is applicable to human health in any kind of near term, medium term type of scenario? So I think it's a really interesting origin story in how you approached it, as well as being flexible with your plans, right? You were planning on going to industry and figuring out a different problem to solve, but it turns out that this one that you were doing, sounds like, as an exploratory approach, ended up being something that you realized you could wrap your arms around and actually build a company around.
Yeah, and actually I want to just double tap on two parts of that, which I think are important. One is, I'm motivated to help people, to help patients, but I think it's really helpful to focus on transformative impact, because it's so hard to do anything in science. And likewise, it's so hard to build a company. It takes so long. So really, like, making sure that problem matters is helpful. And then that's what I was looking for. And looking back, I think it was a really key part of the success that we've had, because people are really motivated by that. And so not only do people pay attention to it when you're making progress, but people are motivated to join you so that you can make that progress. So looking for the really hard problems are the ones that can be really impactful when you do solve them, even though it might be harder. Actually, solving as a group might be easier than doing something that doesn't matter all that much. And the second thing is, it also fits with the strategy I mentioned before of choosing to work on something where you can uniquely make an exceptional contribution, because it's really valuable to get out there talking to folks, asking about the problems they have, telling them about the progress you've made, learning where that might be in the right direction or the wrong direction. And if you're shy about that and don't want to share too much because maybe someone's going to steal the idea or run with it, then you're not going to learn, maybe the key things that are going to put you on the right track. But if you're exceptionally qualified, you're the best person, you can move this faster than anyone else, then no one's going to be able to run faster than you. Or if they want to help, they're going to want to join you, right? Or contribute in some way to this effort that is being catalyzed. So it's a good combination of things that enables you to learn quickly, and to do that without much concern. You still have to be thoughtful about what you say, but you don't have to be secretive about the things that you're working on, so that you can get to meet the right people and learn from their expertise in a way that doesn't have to be through trial and error.
Yeah, and that's an interesting point, and I think maybe a counterintuitive one, perhaps, for some in our audience. So many of our audience members are, entrepreneurs, nucleate activator members, etc. So it's interesting that you're mentioning that if you are transparent because you have, leaps and bounds ahead, competitive type of advantage, or unique insights where you can move faster than anyone else, then you really don't have to worry about sharing, information, obviously within a certain realms of boundaries, right? Like, you're going to have some that are truly confidential and sensitive, but that you can share quite a lot, and that you can gain more insight from people who can potentially help you if you are sharing more information, so that they can guide you along your journey, provide insights.
Yeah, absolutely.
Great. And so, you covered your origin stories, and, the connections with, George Church and Harvard, but yeah, curious about the resources that you used from Harvard as well as Lab Central, which I know is one of the key resources early on in your journey, and curious how these resources impacted your journey at Dyno, and any lessons for aspiring entrepreneurs based on the learnings you got from those resources.
I mean, certainly finding places where you can meet the right people, because those connections, while they might start accidentally, like, they really help you to solve problems, and you have to invest in building those connections before you find the problems. That's always much better. So I think both, that was certainly true in George's lab and at Harvard. At Harvard, I was at the Wyss Institute. That's where our portion of the lab was located, and the Wyss is they're really focused on applications of the technology and, even spinouts. So they have different folks who can help think about the business or help you think about IP. So just telling them what you're doing and asking questions about their jobs and trying to learn how others have had success in the past, those are great resources that I benefited a lot from at the Wyss. Lab Central is also great because it's a very scalable model. You can start with a single bench, but with that one bench, you get access to all of the lab equipment. So you don't have to make all those purchase decisions. You can, use things that are pretty good. And then where there's a few things that you maybe don't have, just focus your resources there and then scale up from like what we did, one bench to two bench to a few benches to a suite. And then eventually, we graduated from Lab Central. The other thing that was really great about Lab Central was they just have a really great community and culture. So there's lots of other companies there also working on problems, sharing in that journey. The space feels great. There's a lot of energy. So when we eventually graduated from Lab Central and moved on to look at other spaces, and ultimately we built our own facility, a lot of the things we liked about Lab Central came with us. Sometimes the things were set up in the offices, but even other times like the exact equipment that we had used at Lab Central, that was pretty good. So we'll start there. So call them platforms, and thinking about like which platform you're going to build on can really help, because it's hard to figure everything out yourself, and maybe you don't want to reinvent all of that. Nowadays we have another collaboration with NVIDIA, and we're building on one of their platforms, BioNeMo, similarly, because there's a lot of things that they've figured out that enable us to deploy our technology, getting in more people's hands in a scalable manner. So choosing the right platform is really key. And these are great partnerships. We benefit a lot from partnership with Lab Central, obviously from working with George and with Harvard. And it's a great way to set yourself up for growing fast when you found the right opportunity.
Got it. And what's interesting about that theme that's kind of been throughout is that you're working with similar mission-driven people, and also that the people help you solve problems that they may have solved before, or leveraging experience, or even if it's another entrepreneur working next to you at Lab Central, for example, that they may have gone through a similar or related challenge that you can learn from. And I'm sure similar stories with the Wyss Institute as far as solving scientific problems. So I think that's an interesting through line from your experience in terms of people who've helped you and have solved similar or related problems. And so you're not starting from scratch, you're starting from a related starting point and some sort of story or experience from someone else that you can leverage as you build your company.
Absolutely.
So I don't want to go too deep into the technical side here, but at a high level for our audience who is not very familiar with gene therapy, can you give a quick overview for adeno-associated virus gene therapy delivery, as well as the machine-guided design approach that Dyno is using to improve delivery?
Most people will be familiar with different genetic diseases. For example, DMD is due to a mutation in the dystrophin protein. So we can help patients who have those diseases by either adding a functional copy of the broken gene or by editing the genome to fix the mutation. And that's maybe the most simple form of gene therapy. For example, editing or addressing a monogenetic disease. But there's also potential to use gene therapy for many other diseases, even non-genetic conditions, because you're adding some new function. And so there's so many different functions from the proteins which are within all of our cells. There's all sorts of new ways of applying those functions, or new functions that could be added that could put cells in a healthier state. So that's all at the genetic level. The big challenge has been, well, it's a beautiful idea, but for many decades, finding a way to deliver the genes into the right cells safely and effectively has been challenging. And that's where the discovery of AAV capsids led to great progress. And today for in vivo gene therapy, that's where there's the most activity, because it's the most effective way, both in terms of the amount of genes you can deliver and in terms of the cost. It's the best thing we have, but it's not good enough. So we're not able to reach, in some cases, enough cells to have a therapeutic impact on a patient. And even in those cases, it's often very expensive to produce one dose of a gene therapy. It might cost hundreds of thousands of dollars to make a single dose. So that makes the therapies even more expensive. The potential, though, is really high. So one, the diseases, they often have very high unmet need. We understand them, we can diagnose them, but there's just no way to treat them unless you can get into the cells and deliver a genetic payload. So there's great reason to want to do this. Quality of life change for a patient can be transformative. In some cases, these are deadly diseases. And, for example, one of the FDA approved gene therapies is Zolgensma for a disease called SMA, and that's often deadly in the first 2 or 3 years of life. Was the leading cause of childhood death due to a genetic disease. And this gene therapy reverses that, and kids have really transformative recovery from that condition. So the potential impact is really clear. And what we focus on at Dyno is we want to solve delivery, meaning with this platform of AAV capsids, can we engineer them so that they can be much more efficient for going into all the different cells in your body? There's never a reason to do that in nature. The virus maybe just needs to go to anywhere where it's easy to replicate. So we need to engineer them to be more efficient for going into muscle, or more efficient for crossing the blood-brain barrier and delivering payloads to every cell or to every neuron. Those are the types of problems that we're working on now. And again, people have been working on that for many decades without as much success as we needed. Capsid protein is really challenging to engineer. You make even a single mutation and it can break all function. And so if you want to make multiple mutations, like to get a gain of function, crossing the blood-brain barrier, for example, it's really hard to do with a random approach. So the thought that I had, again, going back to those technical insights, was now we can make all these changes in a library, and then we can measure out what happened in a high throughput format using DNA sequencing. Often that's done with a DNA barcode linked to the gene, and then that's all packaged in the capsid. So that's how we get the linkage between the barcode and the protein function. So we can now learn how to engineer capsids much, much better. And then with machine learning, we can automate the analysis of that data, find patterns, build models which are predictive of success. And then we can query those models. We can test billions of sequences in silico and find the sequences that the models predict will be improved. We do all sorts of checks on that to rule out false positives. That's really where the art of it comes from. But then we can test those predictions again in high throughput using these oligopool technologies for DNA synthesis. And then we can repeat this process iteratively. So that's what we call machine-guided design. It has the potential to be much, much faster than a random approach, like a directed evolution approach. And we can also transfer learnings from one domain to another much more easier, because we're doing that not just at the physical level of the molecules, but at the computational level. We're taking the information out of the molecules, and then we're putting it into our computer, into our database, and then we can bring that information back into a new experiment, transfer that learning in towards another application. So seeing how you could apply that technology to each of these different capsids, engineering them to be better for different cell types, different organs, that's the reason why I felt Dyno really would be an amazing platform company. And because we get so much return on every investment we make in the platform. And furthermore, every time you improve delivery, you open up new opportunities for new therapeutics. But if you further improve delivery, that's going to continue. So as we bring the cost of delivery down, first we're going to make it possible to treat all sorts of new diseases, and then bringing the cost further down is going to make that affordable, ultimately to the point where it could be affordable out of pocket. And now the potential of gene therapy could be reaching everyone. And it feels like we're early days when it comes to how to apply AI to protein engineering. So seeing this technology wave coming made me want to find some structure that was going to enable us to continuously innovate and achieve this broader impact for genetic medicine.
And that's a really fascinating insight for how you're using DNA barcoding. And sounds like you're using a more directed approach to designing AAVs. And you alluded to this difference between a kind of like random spaghetti against the wall approach, more type of like random editing approach versus what it sounds like you are doing at Dyno, which is more informed, hypothesis driven. You can identify specific parts of the capsid that you want to design or edit in a more directive way. Is that fair?
Yeah, absolutely.
So that's really interesting that you're going to be able to design, more affordable capsids. It sounds like you're going to be scaling up your manufacturing approach, that you're using tissue tropism in terms of how you're designing the capsids to focus on, say, delivery to muscle or brain tissue, etc. I find that really fascinating as you look forward into the future. So you mentioned something interesting in that last discussion about Dyno being built around as a platform company. For our entrepreneurs in the audience, many of them are working on broad technologies that can have applications as a platform or building their own pipeline. I'm curious if you had a moment like that when you decided Dyno was going to be a platform company, or if that was the intent all along on how you approached it.
So I saw the potential all along. And you're right, this is like a classic biotech strategy question. So I guess two things about that. One is I did the case study at HBS, asked the experts what they thought. Everyone told me just do the standard thing, right? Platform great, but what's your lead indication? Like, how is this going to fit into the typical mold for a biotech? So I got the advice early on to not focus on platform, focus on the therapeutic product. But I felt like there was something different here. And it took me some time to like understand what the difference was. Ultimately, I felt that capsids are one of the most amazing platform opportunities because the payload is modular with the capsid technology. So you can put many different payloads in, and therefore you can make many different products. Anytime a product is successful, two things have to happen. The delivery has to work and the payload has to work. But because the delivery is part of that, now you know that it's going to work for all the other payloads that you put in. So there's quite a lot of value that's created with the first product. And therefore it works as a platform maybe better than if the output of the platform, each molecule is its own experiment, and you're not able to transfer learnings from one molecule to another, which is how more biotech platforms work. Call them like a product engine. So there's something more platform-like about capsids because of that potential to reuse the molecules across therapeutics. So I really like that. And furthermore, when I was talking to other companies about these problems, they were very excited to use other technologies. So it seemed like there was a pull. They were looking for ways to solve the delivery problem, and in some cases trying to do it themselves, but still looking for solutions externally. So there was definitely a lot of customer demand. And in the end, I felt like this would be in the pitching to investors, like I need to make a more clear point around why this difference is important. Of course, it's an opportunity to do either one of these, but we think that the platform approach is the bigger opportunity. And more I've come to, so maybe that was the first 4 years while working in the academic lab, not sure about how exactly I was going to spin out the company. But then in coming to that stage where the technology was working, refining the pitch, seeing that this was a tension, and then just trying to address it really directly. Here's why we're going to do it this way, not that way, even though it's different. That was really pivotal. It's also a good filter because ultimately we want, as part of our team, which includes our investors, we all want to be in alignment. So being really upfront about this difference also helped me to find the investors who understood that model and are on board with it. Whereas it might have been harder to get everyone involved and then to find out later that we don't all see the opportunity the same way. So the biggest opportunities are non-consensus and right from an investment perspective. And so I realized a way to take this difference and turn it into a feature of the opportunity. And for the right investors, they would see it that way. And then it would be even more attractive to them. And I think part of that is that, gene therapy has gone through ups and downs, but at times it has been very trendy. And likewise with machine learning and AI, it goes in cycles. So at one point I was telling people about what I was working on for 4 years, and they would say, "Oh, that's great! It's the two latest exciting things combined together in one pitch." So I was looking for ways to make the pitch a little less expected, right? To make those differences more obvious, and then find the right people who are going to support us. Finally, I guess like where we've gone from starting the company to now as a platform, We need great technology that's going to solve problems that our partners care about, but we also need to be really good at partnering. So I realized there was also an opportunity to be excellent at partnering in terms of how we approach our partnerships, how we help them, how we share information, how we work together as one team to really build trust, to really build a relationship. So, for example, we work with partners, and rather than calling that group alliance management, we call it partnership success, just to make it really clear what we are aiming for. And we've got different principles like that we're all working on one team that help us to align with our partners and ultimately overcome all the challenges that we face doing really cutting edge science. So for me, what I'm really excited by is that not only am I going to work at the cutting edge of technology, but we're also going to be a really great partnering organization. And, not just based upon technology, but also based on our reputation and our values, that we're going to build these relationships which really last a long time. And together with our partners, we're going to make the world's best gene therapies. And that's what's going to be the key to helping more patients.
That's great. And informative too, that when it came to deciding, the classic problem of platform versus building your own internal pipeline, you're looking at the fundamentals of what you're doing, right? Like you're building something that can apply to many, many different gene therapies. Probably it would be difficult to internally develop all of those different tissue types of gene therapies, the separate types of clinical trials that are required, the different commercial organizations to build, say, a neuro-related gene therapy versus a cardiac versus a muscle, like you alluded to. All those require really different levels of expertise. And fundamentally, your technology is a true step in improving AAV gene delivery, like you talked about from the beginning of the podcast. Like your true innovation is in delivery, and that approach can apply to so many different areas. So from a practical perspective, if you're going to have the impact on the most patients, which has been kind of your byline throughout this, conversation, that if you're going to have that kind of impact, that you're going to need to partner with people who are, experts in their specific tissue areas, disease areas, and have those resources. And that you did a lot of work early on, understanding what problems people are facing in pharma and with biotechs, and understanding investors and the investor types. So you were able to make a pretty well-informed decision that you really took your time early on with customer discovery. You really understood deeply your technology and the application of it. And it sounds like from that, you were able to say, okay, I want to do a platform approach in addition to the business model of partnering, which, definitely want to spend more time on. But is that a fair assessment that, you really did that early to customer discovery work, and it was really about the fundamental application of the technology, that you want to develop as many different types of gene therapy as possible with your platform?
Yeah, absolutely. So what I really didn't want to do was solve the wrong problem, because that, it'd be so much worse. Working on the right problem and failing, you're still maybe one step away from succeeding. But if you're working on the wrong problem, it's a lot harder to navigate to making an impact. So that's why that model that I mentioned early on around trying to understand the needs and really find a path to making something people want, that was really important to me.
Yeah, and I think that's honestly one of the big lessons for entrepreneurs that I think is unique from your experience, is that you really spent a huge amount of time, right? You spent 4 years as a postdoc in George Church's lab, not only understanding the technical side of your problem, but really understanding the market fundamentals, like understanding what problems biotech and pharmas were dealing with, what problems patients are dealing with, and how they can be solved with your platform. So I think that's a really unique aspect about your founding story and Dyno's founding story that are unique, that you really deeply understood, you know, the patient problem as well as the business problem for how do you approach these patients? How can you actually make a technically really fascinating solution actually be practically making a difference for patients, and really addressing the key issues that, the market stakeholders in between, the technology developers and patients that actually develop and deliver the technology to patients. What problems do they have, and how can you fundamentally solve those with your novel approach? I'd like to touch on more something that you brought up about partnership success, what colloquially I would term as business development. So fundamentally, you have a really interesting, successful technology so far, engineering new delivery approaches for AAV gene therapy. And to the point that we got to, right? You have the technical approach. Ultimately, you want it to get to patients. And in between, you need to partner with organizations that have the ability to develop individual drugs, right? So for each of your capsids, you need to have a, payload, or for people not as familiar, just a therapeutic, ultimately, something that treats patients, needs to be encapsulated in that delivery vehicle. It's critical to Dyno success, right? Like you develop a delivery approach, and you need someone to develop it into a drug ultimately that gets into patients. So I want to talk a little bit more about how you approach those partnerships. You talked about partnership success. You've had several successful partnerships with Astellas, Sarepta, and Roche. Just curious, when you're thinking about partnership success and your partnership success team, what have you learned about these partnerships? What have made them successful? How can an entrepreneur think about those types of partnerships proactively like you have?
I mean, I guess I approach it from two sides. One is relationship building, and the other is problem solving. On the relationship side, we knew as a small startup that we couldn't partner with everyone at the beginning. Ultimately, I want to do that as part of Dyno's mission, but in the beginning, we had to be selective because it takes a lot of work to make any partnership successful. So, looking for folks who were really committed to the modality and who were experts at it, because with this partnership model, we only succeed when our partners are successful and when patients succeed. So working with folks who had a really deep experience was important to me because that was going to better ensure our path to patient impact. And also because I wanted to learn from that experience in a way that would inform where we applied the platform and then set us up for future success to partner with others. So that's why, for example, partnering with Roche also acquired Spark while we were in discussions. And so Spark developed the first FDA gene therapy. So there's deep experience there, plus the experience that Roche has built. Similarly with Sarepta, similarly with Astellas, those partners are really committed over the long term to gene therapy. And it makes sense to build a relationship. We decided to start in a high priority area. And that's a great way to partner because you can start somewhere where it really matters. That's where resources are going to be applied to make sure you move things forward. And then once you start to be successful, then you can think about how do we build on this foundation that we built and expand the partnership. So for example, with Roche, we very recently did a new partnership, which is more expansive also in the CNS, just because we were so excited about the progress that we had made from the first one. And then I guess the second part of it is problem solving. What I thought about for those early partnerships was, given all the technical uncertainty, given that we haven't yet made the capsid that we know our partner is going to use, that's some years ahead. The technology has a lot of potential. We know how to measure the right things. We know how to optimize them, but it's still going to take time to apply it to make the right molecule. So how do we design the structure in a way that enables us to learn how it's going, to check in frequently enough that when we have succeeded, we can move things forward quickly. But when we haven't yet succeeded, we can take that information back into the work and adapt it. So the way I thought about our early partnerships was designing them to be longer term, to be flexible, so that we could ensure that no matter what the technical challenges were going to be, there would be a path to success for the partnership. Now that we have made a lot more progress building capsids, measuring their properties, demonstrating really transformative improvements, say for muscle delivery or brain delivery. Now we're more sure about what's working. So that enables a different type of partnership, still with the goal of always enabling partnership success, but the risks are different. So we can take a different approach to get there faster. So I think that principle of every partnership is going to be successful, and we're committed to that. And then building a strong relationship so that we can navigate that journey together, foundation of trust. Those are two things that have been really key to the success of our current partnerships.
That makes a lot of sense. And it's interesting that you mentioned that your earlier partnerships did have some fundamental differences than your more recent partnerships now that you've had more of a track record. So when you're thinking about earlier stage entrepreneurs, making those longer term, more flexible partnerships sounds like it was key for giving, both sides the flexibility needed in order to get to success. Since, to the point that you've made a little bit before, is that science is somewhat unpredictable, and that it takes time to get to the end goal, and that there might not necessarily be one straightforward path to get there, that you need that flexibility in order to explore the potential options and pivot if something fails, and approach the technical problem in a bunch of different ways. So I find that part really interesting, that your early stage partnerships differ since you've gained more validation of your technology. Any advice for an entrepreneur, besides that flexibility in long-term partnerships? Any advice for building those more early stage R&D collaboration type of agreements that you may have had in the past?
For entrepreneurs who are coming from a scientific background, I actually think that scientific method's pretty applicable here. So scientific method is you have a hypothesis about the world, you do the experiment to test that hypothesis, you gather data, you evaluate and prove or disprove it, and now you have a different model of the world, maybe leading to new questions, new hypotheses that you then go and test. In business, you have a hypothesis about where value can be created. You need to gather the resources to test that hypothesis, just like you were going to do an experiment. Maybe that's funding, maybe it's other things. And then you do the experiment and hopefully in this case have demonstrated some value. Now you've increased the value of the company. Now you can both have a more clear picture about where the future value lies, and then you can form new hypotheses about that and gather more resources and go out and test them. So there's this iterative process to that, this like cyclic process of the method as you're navigating to create value. And then the only difference with the business side is that you're adding in this resource component. That could be funding, could be technology, could be people, and just thinking of those different dimensions. How do I get to the next stage where I have a more clear picture of what the value is? That's the only difference. So for me, when I saw that connection, it really helped me to strategize. I don't need to solve all the problems right now. I just need to get to the point where I'm more certain about where the value is, or where I've definitely created some value. And then that success is going to lead to more interest, more investment, more people wanting to come join the mission. And then repeating that over time. Every time you're adding a little bit more, and over a long time, that really adds up to a really big impact.
No, that's great that you were able to leverage your mental model from the scientific research world to business development, which is one I actually hadn't heard of before. So that's a really interesting and unique approach. But I can definitely see how being hypothesis driven for what experiments you run and how you add value to your partners makes sense for both sides.
It makes it easy to pitch. Like as a scientist, I don't want to overstate things, but I can say like, look, here's the hypothesis. Here's what we think is true. Here's why. There's a pretty good chance this is true. If it is true, here's what it means. Here's the upside. It's amazing. If it's not true, we're going to find that out in this amount of time. It's going to cost this much. And, here's the backup plan.
Mm-hmm.
Investors can look at that and they can play the odds. They can say, yeah, the expected value of this is good and it's worth this much, and I'll put the money in. So math just really checks out. You don't have to discount the risk. And that actually makes it easier because not everything is going to work out. And if you're upfront and honest about that, then those investors are still going to be willing to back you for the next thing, right? Or maybe you learn something because of that and they understand how you got there, and now they're even more excited. So there's lots of different ways to pitch, but this one coming from a scientific background, like I felt was compatible with scientific principles, and maybe even a good filter for finding the right investors who understand the challenging nature of the work that we do. And but still making sure you point to the upside. You have to find a way to connect the impact back to the dollars for your investors, because they have a responsibility to their LPs that they're going to return on that investment. So got to make sure you make that crystal clear, but then you can do that in a way that accounts for the challenges that you're going to face along the way.
Absolutely, that makes sense. And speaking of challenges you faced along the way and thinking ahead for how you make an impact for patients, what's the future for Dyno when you're looking 10 years into the future? You said, you want to use gene therapy for more than just monogenic diseases. Curious about any, key themes you're thinking about as you look towards the future.
Well, I'm really excited about our current partnerships and the work we're doing there, and really looking forward to the technologies like our capsids becoming part of gene therapies that are going to go into clinical trials and help patients and ultimately be approved and out there on the market. I think that's still going to take some years, but it's super motivating to me and our team, and also really exciting to see that we're making progress towards that. I also think that gene therapy is at a really critical time. Again, it's been like a 40, 50-year journey to get here, but we're starting to see it work. And all the different components are coming together. And even where there's challenges, if you look into why there's a challenge, there's often a solution that's already here. It just hasn't yet made its way into the clinic. That's true about delivery. It's true about other aspects of manufacturing. It's true about how we design the payloads. It's a really amazing time of innovation. And when these pieces all come together, that's when there's going to be really transformative impact for patients. And then finally, what we need to do there is we need to bring the cost down to make it so that everyone can benefit. So those are the ways that I think about our platform, is how do we not just solve delivery, but then make full use of that opportunity. It's really, the first gene therapy is working. It's a big step, and it really changed for me how I thought about medicine, because all of us are born with a certain genome, and all of history, that's the genome that you die with, right? There's no change. But now with gene therapy, we're making changes to the genome. And so now there's decisions you can make for how to be healthier. There's agency over that. And that's a really big change. Even though I think we're just taking the first steps, over time, I see lots of ways that that can be really transformative for our health. There's so many things that we know now about biology, about proteins, how they work, how to connect all the pieces together. And so applying those learnings to make ourselves healthier, to put ourselves in a healthier state, we're really just getting started there. So I think monogenic diseases, especially the ones that are serious or that have no other good treatments, that's definitely where our focus is going to be. And it's been a long journey to get here, but over the next 10 years or even the next 20, 30 years, so much potential for what we could do with that technology once we can get it into cells and once we know how to apply it. I'm really motivated by that. And that's why we made it Dyno's mission to work on these high performance genetic technologies. Partnering is the way that we're going to get that technology out there, helping patients through all these collaborations and partnerships, but really advancing the state of the art in that technology for many decades to come is something that's really exciting for me and also for Dyno's team.
Fantastic. And on that hopeful note, thank you, Eric, so much for joining the Nucleate podcast today.
Yeah, thank you so much, Sam, for having me on.
Thanks for listening to the Nucleate podcast. Be sure to subscribe if you'd like to keep up with future episodes. See you next time.